All right, we're going to start our class. Welcome back. Here we are on Thursday. Yesterday I missed school because I had some kind of food poisoning. I know that I think it was food poisoning because another person that I ate with got it too, and just completely wiped out. And so I'm kind of wiped out today. And so if I stumble on my words, I feel fine, just really kind of tired from that experience. And, again, I'm pretty confident that I'm not, sick with a disease you could catch. So we could still use some more applicants for this internship program next school year. I mentioned a couple times before in class. The deadline was last Friday, but if you're interested in doing it during next school year, you can still talk to me about it. All right, and here's what we're doing this week. We're following up on exam 2. And, wow, this is a hard course. And it's hard, I think, because there's just so much information. Each individual piece, I see students nodding, and I think it's understandable, but it's just a question of, absorbing all this information as we go along. And so it's really important for us to follow up, as I described on Tuesday in class. So really look at that, follow up with me. I've talked to a couple students, and you can have a pretty good handle on where you're at when you look at the answer key video. I attached the grade distribution so you know the letter grades that you've gotten on the first 2 exams. And they're worth a big part of our course, but there are more points ahead because of the reality of students understanding just what the expectations are in the course. And so we're rolling forward to our 3rd exam, which comes up really quickly. And the 3rd exam is going to cover this chapter on the nucleus that we're talking about, then a chapter about protein synthesis, and then a chapter about the cell membrane. And so we're talking about the nucleus, so look at that chapter. Then our next reading is going to be chapter 11, protein synthesis. And I put this in red because I added an assignment. So on Tuesday I said, we're going to have a homework assignment on RNA processing. And then I have the video, but I haven't posted on Brightspace yet. And I realized that it would be best for us to do a different assignment first. So the due date for the RNA processing assignment has moved down. And we have a different homework assignment that's due 1 week from today. And it's a protein synthesis assignment that's really the biggest part of chapter 11. So we're going to do chapter 10, nucleus, then we're going to talk about protein synthesis. And with protein synthesis, you talked about that in intro bio, and it involves the ribosome and amino acids and stuff like that. That's actually a different part of the story of protein synthesis that's covered in this chapter. And so we've got a homework assignment, and it's going to have these descriptions, and it's a very straightforward assignment, but it's very good to see it as a package, because I just talk about the synthesis of 4 proteins, and in talking about those 4 different proteins, explain most of chapter 12. And the assignment is just to describe how is this protein synthesis, and the proteins are hemoglobin, insulin, PDGF receptor, and then something called the lysosomal proton pump. And by describing those 4, and each description is very similar. There'll be just one step that's different like that, right? So that's going to be due a week from today, and then the other one a week after that. So this one is now due 2 weeks from that. And like I said, I haven't even posted that one. That RNA processing one is, I described before, it's what happens to the RNA before it gets to leave the nucleus. So we've learned so far in our course, and the assignment starts with that, here's the DNA, here's the transcription unit. We're going to transcribe it into RNA. That's called the primary transcript, and before that transcript can leave the nucleus, it goes through this RNA processing. And so that's dealt with in chapter 6 in our book. you don't have to read and look at that chapter ever, but because it happens inside the nucleus and it's really nicely connected to gene expression story, I made it into a homework assignment that fits with chapter 10. But again, that is, so this protein synthesis assignment is going to be right in the middle of the chapter. It just explains almost, most of the chapter. Then the RNA processing one is quite different than the chapter on the nucleus that we're talking about right now, because it's dealt with at a different point in our textbook. And then we're going to have our exam on 3 weeks from today. And it's going to cover chapter 11, chapter 12, and the chapter on the cell membrane. Maybe that's chapter 14, and all the assignments and so forth. And we're using the examples throughout here. We're using the examples, so we're not asking you to go back and study the material for the first and second exam to get ready for the third exam. But we want to go through the answer key video and be solid on that, because we just keep building up, and we're using those examples as we go forward, like that. So that's where we're at, okay? And I've got the video for the protein synthesis assignment, and I'll post that with, today, like that, okay? And yeah, it's chapter 14. So this third exam is going to be chapter 11, 12, and 14, okay? And so we're inside the nucleus, and we're talking about the nucleus, and we're talking about how to get in and out of the nucleus. We talked about this nuclear pore. The nuclear pore is awesome. It's a gatekeeper. But then, as soon as we do, we say, the nuclear pore, all we have to know is it exists. We don't need to know the details of the structure of it and so forth that our book puts in drawing and puts some labels on. Instead, we want to learn about how things go in and out. And it turns out that we don't even talk much about the nuclear pore. We turn our attention to, if a protein's going to go into the nucleus, it has to have nuclear localizational signal, and it's going to have some helper, important, help it in. And if the nuclear localization signal gets blocked by a protein like 1433, it's not going in, like that, So we're talking down that pathway where we actually, in talking about getting in and out of the nucleus, we kind of turn our attention away from the nuclear pore, and it's these other components that really allow for entry or exit. And so that's a big part of our story, and it's on this pathway of gene expression, because that mature RNA has to get exported using something like exportin out of the nuclear pore. I mentioned, and we'll talk a little bit more about it, that anytime there's RNA in a cell, it's always bound to proteins. And it's actually the proteins that get exported when mature RNA gets exported. And so those proteins change as the RNA is getting processed, the proteins that bind to the RNA, and the mature RNA has a set of proteins that say, I'm ready to leave the nucleus. And those proteins go out along with the RNA, and they're carried out by a protein like exportin, okay, like that, okay? Okay, so we're on this part of the chapter, and that's the bulk of the chapter. We'll go through these other parts of the chapter quickly. Our book goes through them pretty quickly. We'll go through them even faster. So entry and exit, like that. And nuclear localization signal, FOXO, okay? So let's just think about FOXO a little bit more. So we saw, it's kind of funny, because here this is the first day of class, and we didn't mention the nuclear pore. We're just like, get in the nucleus, right? And yeah, the nuclear pore is ready. We don't really need to know anything more about that, because it's all about that 1433. And so we talked a little bit about last time about how 1433 binds to FOXO and covers up the nuclear localization signal, like that. And Sophie asked a question when we were talking about that binding. What does it mean, and what is it like? And I talked a little bit about that and gave a hypothetical example. Well, I gave an example of insulin, binding the insulin receptor, and kind of talked about that idea hypothetically, based on, there are hydrophobic amino acid side chains, hydrophilic. And after class, a student asked me about that talk. You don't need to be able to describe that on the exam. That was just a description of what this binding is like. It's non-covalent interactions, and it would be between the amino acids on two proteins, like that. Okay? And I just want to go back to that, because there's one thing I didn't mention that's good to mention about that whole binding story. And If we just look back at 14.3.3 binding to FOXO, another student asked the question of, does 14.3.3 always bind to phosphorylated targets? Because that's what's happening here, right? When FOXO is not phosphorylated, it's not bound by 14.3. The key is it gets phosphorylated by AKT, and then suddenly, oh, it has high affinity, right, like that. And so the answer to that question is, I think, yeah, pretty much always 14.3.3 proteins bind to phosphorylated targets. Those are called phosphoproteins. We haven't used that term here, that's fine. And it looks like that's the general rule. And so just to go back to, again, just that talk and thinking, you can see, just by the way the artist drew 14.3.3, that 14.3.3, as part of its binding region, has these pockets that are waiting and hoping to find a phosphate. Phosphates have a lot of negative charge, so we can anticipate that here on the 14.3.3 binding surface, there'd be lots of positively charged amino acid side chains that are going to be just right to match up with the phosphoprotein, like that. And that probably the other parts of the surface between FOXO and 14.3.3, they probably have some hydrophobic amino acids, so like the example I gave, where they would like to press together. But that's pretty non-specific, that can come and go. But if you have a hydrophobic surface with a couple of really negatively charged areas because of the phosphate, and you have positively charged on the 14.3.3, you get this thing that's like a salt bridge where water gets pushed out and you have a really, really stable hydrophilic interaction between the positive and negative charges like that. Just like how with table salt, it's really a strong crystal when water is excluded. You add water, falls apart right away, like that, yeah? And so, see, that's where you get that high affinity and specificity is, oh, well, it doesn't bind to FOXO because it just got these sloppy, weak binding sites, and it's not matching up with those positive and negative charges on the 14.3.3 and FOXO. That would really increase its affinity, like that. Yep. And there's just lots to say about it. It's, that's down at that, protein structure and function level, right? Because another thing to say about that is, right, FOXO can get activated by removing those phosphates, but it's kind of tricky to get them removed, when it's bound to 14.3.3. So 14.3.3 is acting as a molecular switch in a couple of different ways. And one is keeping it out of the nucleus. The other is protecting that FOXO from getting dephosphorylated by a phosphatase, because how are you going to get in there and get it done, like that. So, it's really interesting to see, and we've talked a little bit about this course, about this idea of a molecular switch. Turning something that would be kind of like a dimmer, a light dimmer, into a crisp switch. We talked about that at, cell cycle. It's starting to flicker, and we really want it red or green, like that. And so down at these levels there, you can just see, oh man, that's a really satisfying interaction. The thing I didn't mention when I was talking to answer Sophie's question about binding is another thing that happens when two proteins bind together, like when the insulin binds to the insulin receptor, when they bind together, they almost always change their conformation, change their shape a little bit. And that just, they're built to have a little, almost like a teeter-totter shift in shape, just a little shift in shape, so they match up well. And here's a good, and not to confuse anybody and everybody, but here's a good example of that. Here's a good example of that, just to be aware of how they change shape. So we think, okay, what we see 14.3.3 family members doing, and they do it a lot. We saw last time it's a family of proteins, and they're involved in just about everything a cell does, one way or the other, like that. Okay? So that's their plan, like that, right? And what they do almost every time is they just physically block the nuclear localization signal, right? You could point to FOXO's nuclear localization signal, it's under there, like that, okay? Okay, so I don't want to confuse people, but it turns out that this happens sometimes for specific switches. This happens, and so I'm just going to draw this just because you see this some, and so we could just draw this, sort of generic 1433, and it looks like this, and then we could say, FOXO looks like this. And when they bind, they're going to shift their shape a little bit. So FOXO is going to maybe look a little bit more curved like that, and the 1433 is going to be a little more curved like that, like it's kind of wrapping around it, sort of like that. So that almost happens, always, like that, okay? So that's our story with that, and again, it's because now we have that phospho on the FOXO, like that, okay? And so that's the typical thing, okay? So now I'm going to try to draw this, and this is the thing that I don't want to have people confused, but just so you see this. So sometimes it turns out that when 1433 binds to its phosphorylated target, so we'll use a different kind of color here. So here's a target, and we'll draw it differently just to show you that when they bind, they're going to, again, do that curving. And this one is going to curve like that. And the 14.3 is going to change its shape a little bit just as a natural part of the binding process. And so again, that's a general rule for protein-protein interactions, is they're both going to change their shapes a little bit like a teeter-totter mechanism. And here's the confusing thing that happens. So for some specific proteins, and none that we are learning about in our course, and none that you're going to be asked about in an exam, but just to show you this, for some specific proteins, when 1433 binds them because they get phosphorylated, for that green protein, it actually exposes a nuclear localization signal, and that's how it gets into the nucleus. So that's confusing because almost always 1433 keeps it out of the nucleus. But because the protein the 1433 binds to is changing its shape, there are a small number of specific proteins that change their shape and actually expose, on that other side of the protein, expose a nuclear localization signal, and that's required for them to get in, right? And again, that's confusing, but hopefully, yeah, Bryce?
Are they also blocking nuclear signal?
No. So in this case, the green protein here is stuck in the cytoplasm because its nuclear localization signal is covered up, and it needs to be phosphorylated to bind and bind 14T3 to get its shape changed a little bit to expose it. So, that's one of these examples where it's nice to hear, that's kind of nice. The exception helps us remember that almost always these proteins are changing shape some. So don't be confused by it, because normally 1433 just blocks a nuclear localization signal. So without binding to 433, FOXO gets to go in, because there's its nuclear localization signal. But in some rare cases, and actually some very important proteins, they're like, I can't get into the nucleus because my nuclear localization signal is covered up. I need somebody to help me open up this protein and let me in. Like that? And 1433 can't do that. Yeah, Liana?
Can you give an example of the first, just so I don't mix it up?
Yeah, I think there's a protein called myopodin. Just kind of making that up though too. And don't forget that I'm recovering from a thing where I was, like in bad shape. So I can spell that a little better. You should just be, you know what? You could just, it used to be kind of okay to be Googling during class, to be skeptically checking the instructor. But with that AI overview, it's just wrong, just totally wrong, like that, And, right, that's the next step in education, right, where, one of you start standing up and going, I can Google faster than the instructor, like that. Also, I'll be teaching it. But yeah, But it's, you know, it's got to do that to get in like that. So just to help you kind of, oh, that's a memorable example, because those proteins are always kind of changing shape. And it's just part of it's the natural chemistry. The proteins are built to change their shape. I like that. Okay? So just following up on that a little bit. Okay? And so that story, right? And this is from our first day in class, you know, and we're just like, wow, you know, we could talk a lot about that, right? You know, that could be a figure on the file, and you'd be like, what are some of the things we learned from this figure, right? A bunch of stuff. And so here's this also back near the start of our course. Remember we said, hey, let's make a glowing phosphoprotein and see, you know, if it can go into the nucleus. And we could use that glowing protein as a way to study it, right? So it goes back to that example too, because we can really follow the protein. And what else can we follow? We can follow that 14.3.3 function, that activity, right? We could look at mutations in 14.3.3 and try to understand what is required. We do it pretty quickly because we just look at one of its targets and see whether or not its target gets into the nucleus or not. Yep, so, you know, sort of neat like that, okay? And so just, you know, some slides, because the book is talking about this too. So that nuclear localization signal is surprisingly short, series of amino acids. I like to call it a nuclear localization domain, but it's a signal. It's not really a big domain, a big functional part of the protein. It's just a little patch, and it's just positively charged amino acids, like that, okay? And that's what important needs to bind to. You know, it needs to find that and bind to it. So back to binding, we can anticipate that important must have some chain of amino acids that are negatively charged, negative acidic amino acid side chains that are going to be like, yes, that's unusual to see that much density of positive charge, and I have a lot of negative charge, and we'll match up, and I'll bring you in, like that. Yep. Okay? And so, you know, the book is talking about this and saying, you know, well, you know, lots of molecules, the RNAs, the proteins, they need transport to get to the nuclear pore. Small molecules, they just go in and out, you know, so just some extra information in the book like that. Then we talked about this energy-dependent cycling of both important and exportant, that they need to burn some energy to drive that cycle in that forward direction, like that. Make sure that kind of the door is turning in the proper direction, and that it's an information-dependent pathway, like that. So cool example of that concept. And exportant doing the same type of thing. And again, lots of detail in the figure in the book. We'll definitely want to be able to draw that. All we have to draw is, you know, protein with a nuclear localization signal, important, go in there through the nuclear pore together, then important's got to go back out, so it's going to burn some GTP to get back out, like that. We don't need to, no way do we need to look at the greater details of those other factors. Then an example, the book picks an example that's from chapter 16 that we didn't emphasize, so we would want to pick FOXO and other transcription factors that all need nuclear localization signals to get into the nucleus. Yep. And so that's that. And then we talked some about 14.3.3 and how it's just a dramatically big story. And we talked about this idea, this interactome. Because that's regulating the gene expression. That's post-translational. And so just to skip all the way back to that figure from intro bio, that's from our intro bio, it tells the same story as this. And you know, our course is really focused on transcription regulation as a way of regulating gene expression. But FOXO protein is all the way to here, made protein, and it could still be on or off. And that's called post-translational regulation. And there's a lot of that. And we've seen lots of examples of that in our course, we just haven't labeled it. But you're essentially, you know, turning the FOXO, not the gene off, but FOXO function off when you phosphorylate it, like that, right? So there's lots of regulation along this pathway, specific steps. And so we say transcription regulation, then RNA processing, and then exit from the nucleus, then stability of the RNA, then translation and regulating translation, then after translation, post-translational regulation. And in post-translational regulation, that's how 1433 does its job, right? It's not regulating transcription, it's regulating post-translational activity of target proteins. And in post-translational regulation, we just want to use one other term, and that is, so post-translational regulation means any regulation of the protein after it's been made. It could be interacting with 1433. It could be destroying the protein. That's post-translational regulation of beta-catenin. That gets destroyed. P53 gets destroyed, gets degraded, and MYC gets destroyed. So just degrading the protein quickly is a type of post-translational regulation. Then we also saw post-translational regulation of proteins that need to have a ligand. So the estrogen receptor, that's post-translational regulation. It requires estrogen like that. And so other proteins like that need post-translational regulation. All of those receptors, PDGF receptor, G protein coupled receptors, that's all post-translational regulation. They get activated when they bind to the ligand, the hormone or other molecule signaling factor. So lots and lots of examples of post-translational regulation that are a big story of life on Earth. Yeah. So then we also saw some examples of post-translational regulation where there are covalent bond changes to the protein. So the most common one we saw is post-translational regulation by phosphorylation. We've seen that a lot. Protein get turned on or off by getting phosphorylated. And so that is considered a special example of post-translational regulation. So post-translational regulation right? And and when we're studying for the third exam, we'll be studying for the final. The final is like a week and a half after the third exam, right? We'll have a little bit of new material, but the final week of our semester is we'll be really pulling it, trying to pull it all together, review session kind of thing for any questions you want to cover. So, when you're doing this studying for the third exam, you're also hitting right at the final two. So it's really worth your time to spend this. And this pathway of regulating gene expression is a very good pathway to talk about on a final exam. And that is, okay, so we studied transcription regulation, picked 3 examples of transcription regulation. We described RNA processing, picked 3 examples, of RNA processing. We described, and in the RNA processing homework, there's this RNA interference. That's where the RNA can get blocked and degraded. Pick examples of that. Then, oh my gosh, post-translational regulation. Many, many examples like that. Describe, 4 points on our final for sure. Post-translational regulation. And in that large category, when there's post-translational regulation that involves covalent modification of the protein, so like adding a phosphate or removing a phosphate, that's a kind of post-translational regulation that is called post-translational modification. So a separate term specifically to, we've actually modified this protein. We've physically had a covalent change to this protein. That's post-translational modification. And those are also examples of the larger category of post-translational regulation. And we've got a list of those. Now, with regard to that post-translational regulation, what we did in the first half of our course is we made a list of those with transcription factors, right? 12, 14 transcription factors. How does this get turned on? How does it get turned on? So we really emphasized for transcription factors lots of different ways there could be post-translational regulation. Some of them are post-translational modification. But that's a good place to go to think about examples. There are lots and lots of different kinds of proteins in the cell, and they can be regulated. Our favorite proteins are transcription factors. They're all proteins, and they can be regulated like these mechanisms like that, you know? So that 14.3.3, it functions by post-translational regulation of proteins, like that. That's where that fits in. It's along that pathway. And so the book doesn't really bring this out so much in this chapter, but we want to because it's a cell biology chapter. And cell biology is like, depending on where you are in the cell, you'll have these different categories of regulation. So it's great visual like that, you know, to make a big picture of that and fit those examples in along those pathways, right? And I tried to explain that the big sort of dance of life is, well, these transcription factors, they're gonna regulate gene expression. And so there's a kind of output-input that the genome has set up, and the output includes post-translational regulation of the transcription factors that can involve hormones, and those hormones could be affected by the environment, like some birds that are getting ready to migrate, they see the day length, they change their hormone levels, getting ready to migrate. You know, just huge input from the environment of the cell and the organism and the outside environment to affect gene expression, right? So, you know, it's a good visual that we're getting to. And again, the chapter's focused on the nucleus, but we want to bring it out, you know, to that cellular level for our course to see that big flow of information like that. Okay, super duper. Okay, so that's that. And anybody have questions? And so for that 14.3.3, scientists would love to find out all the different ways that 14.3.3 gets the job done. And they call that the interactome. And they're just stealing the word genome, the sound of that, and saying, we want to find all the ways that 14.3.3 interacts. And so it's hard, because it's protein stuff, so they mention mass spectrometry. There are some methods that we haven't learned about in our course that biochemists use to try to understand all of the post-translational regulation and interactions that occur. And for 14.3.3, they call that the interactome. We would like to know every protein that 14.3.3 binds to. Like that, you know, can we find them all? You know, like that, okay? And that's an example of this post-translational regulation. So that's what that is about, okay? So here's just a little quick picture to show you, and it's just a visual. Don't bother, you know, trying to scramble, write it down. And it's just showing that 14.3.3 in this, it's showing it as a, like that, you know, there's the little thing, and it's binding to targets. And, you know, the targets are just, what? Oh, it's just a long list. So again, don't sweat, you know, trying to see these and know them. But like RAS, RAF, MEC, ERK, RAF is a target of 14.3.3. And in that example, it's not a target for nuclear localization. It has to do with that pathway. And there's BAD, we didn't learn the name of that, but that's one of the pro-apoptotic members of the BCL2 gene family, you know? So it's just a massive list of proteins, some of which are familiar with us. And, you know, when they're released from 14.3.3 binding, they get to do their thing, you know? And so it's a big, big show. And this is just that from that, big chapter review about it. And this is just showing, you know, big categories of cell responses and many, many different stories of 1433. So just to give you a visual of what we heard on Tuesday, and that is, oh my gosh, this 1433 protein family does its thing in, so many different pathways. We don't need to, learn them because we've got a good one, FOXO, very good one, like that, okay? So that's, in and getting in and out of the nucleus, nuclear pore, the organization of it. So then the last part of this chapter is about these. One is this, and I mentioned it before when I talked about starting this chapter, and that is, well, we're really interested in the chromosomes. The DNA is inside the nucleus, that's the big deal. And are the chromosomes organized? And this is cell biology research, and there are just a couple of take-home messages for us from these last two sections. So one of them is, where are the chromosomes? And this is what the data looks like. Here they're stating nucleus to try to see where are the chromosomes located. Now, this would be during interphase, right? Because during mitosis, we can see the chromosomes. So during interphase, where are the chromosomes? Why do people care? That's when the cell does its living. That's when gene expression is occurring in G1 and things are happening like that. It's also where the cells replicate their DNA. So lots of stuff happens to the DNA during interphase, and you just can't see it under a regular microscope. So can we use fancy microscopes, stain it, and make any summary statements? And there's one summary statement we can make, and there's just another example of that. And here's the summary statement. And let's see, do we have a better figure of that? No, this is going to be it. So what this shows is that that when we see the nucleus, there's the nuclear membrane, there's the nuclear lamins, there's the nuclear pores, and then in the last section, nuclear bodies, we'll learn more about this. This is the nucleolus, and this is something that scientists can see under a regular microscope. So during interphase, you can't see the chromosomes. The nucleus in general just looks like a gray haze, but there's a nucleolus, and different species have 1 or 2 or 3 nucleoli, and the nucleolus is an area in the nucleus where ribosomes are being synthesized. So we'll talk about that a little bit more in the next section, but that is a place of organization in the cells. And so here's the take-home message of where are the chromosomes? And the answer goes like this, see, they're trying to draw heterochromatin here. So those spirals, those kind of brown spaghetti spiral, those are heterochromatin. And not right at the nuclear membrane, but near it, we have this open chromatin. And so gene expression occurs kind of near the nuclear membrane, and also near the nucleolus. And that's what they're trying to show. And that's just really the only take-home message we need to get from that chapter, is that the chromosomes are in the interphase nucleus, and some of them the genes are getting turned on, and they're not just getting turned on haphazardly. There's some regions in the nucleus where there's higher amounts of transcription, and there are regions where the genes are more turned off. And so that's a little hint for the final part of the textbook chapter, and that is there are these patches that are called nuclear bodies. And what they're saying there is, yeah, the interphase nucleus just looks like a gray haze, but there are areas of nucleus in the nucleus that have special activity, special focused function. Okay? And one of those areas is the nucleolus. So we're going to make a little list of examples of nuclear bodies, and what that just means is areas within the nucleus where there's high levels of specific types of activity. So we've already seen, we're going to talk about the nucleolus. We're also going to talk about these regions in the nucleus where the transcription is high. Like There's also going to be another region where the DNA replication is high during S phase. And that's what we're going to want to get out of this. Okay, so this is one, and it's transcription. And it's saying that transcription takes place in general in locations, and then as you look more specifically, there are little areas that are like transcription factories, like patches, where that's where lots and lots of action is. And so we see transcriptionally active genes clustered together in patches, like little factories of transcription activity. And that's significant because then the cell biologists are like, well, how do you get to be in that? How do you get to join a transcription factory and be expressed? And how do you get excluded like that? And so there's lots of research about that. You know, is that a way to help regulate activating and repressing genes? You know, let them be in that patch. See how they've drawn a circle around it? There's no circle. It's just a concentrated area. So that's true for all of these regions, is they're not, they don't have a membrane around them or any special structural feature. They're just a high amount of activity. And again, it's in the interphase nucleus where there's lots of important stuff happening. You just can't see it with a regular microscope. So let's just, let me just show you this, that the book has a little table of nuclear bodies like that to talk about some of these, we're going to make a similar table but shorter and a little bit different. Okay, and so let's just start doing that. Okay, so for us, our examples of nuclear bodies are going to include the nucleolus. And so let's just talk about the nucleolus. And the nucleolus is the site in the nucleus of our DNA gene expression. And so just to talk about that, our DNA codes for ribosomal RNA. And if we drew a picture of a ribosome, it would be a whole bunch of proteins made up of about 70 proteins. So many, many proteins make up the large and small unit of a ribosome. So that would be a ribosome with all of its ribosomal proteins. And that's the site of protein synthesis. The function of the ribosome is just to grab a mRNA and start translating it into a protein. It's a fantastically big machine to do that job. And in each of those subunits of the ribosome, there is ribosomal RNA. So ribosomal RNA are parts of every ribosome, like that. And the nucleolus is where all of the rDNA genes are located. So in that sense, it is a transcription factory, but you'll see there's a separate function that is added, and that is that the nucleolus is where those rDNA genes get transcribed, expressed, and they get processed. They get RNA processing right there. And the ribosome subunits get assembled there. So we wouldn't call the nucleolus just a transcription factory. Yeah, a lot of transcription happening there, but also ribosome assembly happens there. And so that's the nucleolus. And there's just so much similar biochemical activity, that you can see that as a region, you know. And again, there's no boundary to it, no membrane around it. In the nucleus, where the rDNA genes are located, is a visible patch of factory work activity of transcribing and processing the ribosomal RNA genes and then assembling them into ribosome, like that. Okay? And so that's the nucleolus. To just fit that in with the chapter and to think about this, so that's where the ribosomal RNA genes, the rDNA genes, are located, like that. Then, scattered on the other chromosomes are the genes for ribosomal proteins. Those ribosomal protein genes, they have to get transcribed. They're not in the nucleolus. They're just elsewhere in the nucleus. They're just proteins getting made. And they have to get transcribed into RNA, the ribosomal gene, and then that would get RNA processing in the nucleus to a mature RNA that would go out into the cytoplasm, where that individual ribosomal protein gene, RNA, would have to get translated by an existing ribosome to make this new ribosomal protein. And the cell has lots of ribosomes. Like an individual human cell has 50,000 to 100,000 ribosomes. So there's lots of work to make those ribosomes. And the existing ribosomes make new ribosomes by synthesizing the proteins. But then, those individual ribosomal proteins must have nuclear localization sites, because they have to go back into the nucleus to get assembled into the ribosomes that then have to be mature ribosomal subunits that exit the nucleus. So one of the big deals of the nuclear pore is regulating these pathways of ribosome production. And there's tens of thousands of ribosomes in every cell, and they're changing the number a lot. So they'll, you know, anytime you say, let's grow, right? So that FOXO, you know, that pathway, we talked a little bit about TOR, and TOR's biggest job is, let's make more ribosomes. You wanna grow, let's just make more ribosomes. Because they make, you know, they are the protein factories, you're gonna make a lot more proteins. So it turns out to be a big, big story. For us, it's that the nucleus is where they get assembled. That's where the ribosomal RNA gets ready. That's where the newly made ribosomal proteins need to get in and get there to get put together into a functional ribosome that then has to leave the nucleus. So for us, for a nuclear body, it's just that. You can see the nucleolus, it's a nuclear body. It's where the rDNAs get transcribed and processed into ribosomal RNAs, and the ribosome gets assembled. Those proteins arrive and they get assembled, and we get functional ribosomal subunits coming out of the nucleus, yes?
So that image of the ribosomal RNA with the ribosome proteins, is that RNA or is that a ribosome?
Yeah, so a ribosome is a complex of about 70 different proteins plus a couple of molecules of ribosomal RNA. And they're together. So every ribosome has the ribosomal proteins plus ribosomal RNA, or rRNA. The ribosomal RNA genes, the rDNA, those would be an example of non-coding genes. It's confusing for students because here's the ribosome, and it's going to bind messenger RNA. That's what you learned from introvio. Its big job is get a hold of a molecule of mRNA, and translate that into a protein. Yep. And you see these rRNAs are different. They are part of the ribosome, like that. And they're not a messenger RNA molecule. Yeah. And so again, our focus right now is, what's our first example of a nuclear body? Oh, the nucleolus. And I mentioned there's 1 or 2 or 3 of those, depending on the species. No reason why there would be more than, well, 1 is enough. Seems like that'd be fine, but just different species do have, and they're closely related species. Like a quail is a kind of bird that has 2 in every cell, and a chicken is a kind of bird that has 1 in every cell. And you're like, pfft. Yeah, but it's a nuclear body, and there's basically 1 or a couple in a cell, in the nucleus, always. And what's going on there? That's where all the ribosomal RNA genes, the rDNA, is located, getting transcribed, getting processing it, cutting it up, getting ready, and then the ribosomal proteins are coming in. Newly synthesized ribosomal proteins coming in and getting assembled and leaving to go out in the cytoplasm and make other proteins. Yeah. So that's our first one, just that. Okay. Then our second one, we can just call it transcription factory. I think people have some other names for them, but when they first started studying them, they called them transcription factories, and that's a good name for us, right, at this point in the semester. And and there would be many of these, and they're just patches scattered, mostly near the edge of the nucleus and mostly near the nucleolus, and they are locations of high levels of gene expression, transcription. And that's all we need to say about that one. Okay. And then the next one we could list that we should learn is called a polycomb body. And polycomb is the name of a weirdo fruit fly gene. And these are locations of gene repression. So there's some areas in the nucleus where, oh, there's a gene that could be on, but it's not. And it's bound by these polycomb proteins, and those cluster together for some reason in regions. So polycomb body is a place where there's a lot of gene repression. And so just another example of that, where scientists see that, and they're trying to figure out, well, why would it be all clustered together? How's that part of the function of polycomb? And we've been talking about gene activation. We've talked about when genes are turned off. There's more, a lot more that a person could teach and learn about how genes are off. And so the first time and last time we mentioned polycomb. Polycomb is a family of gene proteins that help repress gene expression. So that's another one, just to show you, places of activation, places of repression, great. And maybe, let me just check this list again, because maybe there's just one more that we should, that's not good. Let's buy some products. There's maybe just one more. Yeah. Really, there's just one more of these that, for us, we should be, learning about. And that is the fourth one, and that's what we're going to call a DNA replication factory. And that is in interphase. Again, there's no hint under a microscope that the chromosomes are getting copied, getting replicated, that we have DNA synthesis happening. But there is, and there's lots and lots of activity in these patches. And would that mean, like, who would you expect to find in a DNA replication factory? And the answer is, DNA polymerase, that MCM protein, right? And so forth, right? And also we would be origins of replication, right? And during S phase, right, they'd be found during S phase. So only during S phase do you see these DNA replication factories. And again, they're just patches, just areas where that activity is really concentrated. And, you know, just like as the, you know, DNA gets getting copied, it just gets, you know, kind of pushed out of that factor region, and we just keep the, you know, more DNA in the factor region as we're copying it, and we're just like, come on, let's get this done. S phase is definitely a time when the cell has just got its mind on getting the DNA copied. So a good job, we got a lot of DNA to copy, like that. So a DNA replication factor, and that would be it. The book dives into, because, you know, it's diving into some other examples like that polycomb where it's just like, oh my gosh, I gotta talk to you about this, it's so cool, you know? And so you can learn about them, and maybe one that's described in some detail might be a 2-point question. I don't think I mentioned it in the answer key video for exam 2, but I think there were just 1 2-point question on exam 2 that came from the book that we didn't talk about in class. So be aware of that in this chapter, that maybe there'll be a 2-point question that comes out of that list that's another kind of a body, you know, like that. Probably not, you know. So that's always what's happening in class, is try to get solid on class, then go into the book and say, I see this section on nuclear body. I'm not going to try to just flat out memorize every one of these. I'm going to prioritize based on what we learned about in class, try to understand them better from the book, maybe come back to class with some questions about how I'm confused about trying to connect them. And then if there's another one that's important, you know, I'll try to learn that, but that's not my highest priority in trying to, you know, get stuff going. Yeah, that's the way we're learning, you know, for our course. Is our book's really important, helping us understand what, you know, going about in class and stuff like that. Being able to solidify those examples, and then try to stretch out a little bit more, but yeah, it's not like you're on your own. You just learn from the book, right? I had a lot of professors in college who just go, well, read 1 chapter a week, like this book. Our semester has 16 weeks. You see 16 chapters. Go get them. Yeah, all of my professors were over 100 years old. Seriously, it was crazy. It was crazy. Yeah, oh my gosh. And they just did that. I had a professor fall asleep during lecture by leaning against, lectured against the podium, and he just leaned against it and he fell asleep. And then we're just like, we're learning now, man, here we go. Yeah, so, right? So we're trying to help you, you know, grind your way through that textbook, okay? So there it's showing, you know, some of those examples, and it talks about how, here's that one about the nucleolus, and it's talking about, gosh, you know, there's a lot of stuff happening because you got to get those ribosome proteins made out there and get them back in here. You got to get the ribosomal RNA ready and get it out of here. So, you know, there they are coming in, right? There's a big lineup of them coming in because it is a lot. I mean, the amount of production is very dramatic. When you say, let's go, let's get this cell growing like that, it's a big shift in the number of ribosomes. Okay, great. There's another one that's that polycomb body. It's like, we're shutting you guys down. And again, you know, the book gives this false impression that it's a really, you know, an area that's really bounded by some kind of fence. It's not. It's just, you can stain it, and you can see, oh, there's a lot of polycomb. Polycomb is not just smoothly scattered. It's a big repressor. So why is it, you know, speckled like that, you know? Let's figure it out. That's what's going on. So again, not a lot, you know, that's the idea of under a regular light microscope, the nucleus looks like a gray, you know, gray sky. Well, here you go. We stained it with a, you know, polygome, and we see the polygome is really concentrated, you know. So what's going on? That's what that is about, and that's the end of our chapter. Let's see, what's going on here? There's some more with that. Okay? So we have a little bit more time to go here today, and so I'm going to start talking a little bit about chapter 12, and it's this protein synthesis pathway story. So we should pause and see if Bryce has any questions. Bryce has a question.
Is that chapter 10? That was chapter 10?
Yeah.
Okay.
Yep, yeah, chapter 10, I think it's just called The Nucleus. Yeah, yep. And, yeah, Don Diego?
So you said the nucleus is also a transcription factory?
It is, because it's transcribing the ribosomal RNA genes, the rDNA genes. But we should, so it's true, that is happening there, but we shouldn't call it that because it's got these two parts to the story. It's transcribing the RNA genes, the RNA, the ribosomal RNA gets processed there, and then the ribosomes get assembled there. So we want to be careful and not say, oh yeah, it's a transcription factory, because that's not, no one would call it that. They would say, no, it's the nucleolus, it's that. Yeah, but it is, a lot of transcription of those specific genes in that location. Other questions people have?
How would
Oh, yeah, Kayla?
Would something get out of the nucleolus the same way it would get out of the nucleus, like a ribosome?
So no, when we're saying like when the ribosomal RNA that gets processed, that's going to have to get out of the nucleus. The ribosomal subunits, when they get assembled, they're all ready to go. They have to get out of the nucleus. So they go through the nuclear pore, and the assembled ribosome subunit goes through the nuclear pore. I should mention that the ribosome, again, is made up of about 70 proteins, so those would be protein subunits of the ribosome. When you zoom out and look at a whole ribosome, it's made up of two very large subunits. One is called the large subunit, one is called the small subunit. So I kind of drew it that way. And they have to leave the nucleus, this small subunit fully assembled with its protein subunits and its ribosomal RNA. It's got to leave through a nuclear pore like that. And it's cool, the nuclear pores are really wide, like that. They're like, we're ready for cargo, and it's like that, where you're like, oh, dang, I guess we bought a sofa that's too big to get in the door. Well, whoops, like that, right? But that's what's leaving. Yeah. And it needs to have export signals, and I don't know what those would be like. The book talks a little bit about them, but not that. The book doesn't talk about the ribosome exit signals, but when we talked about getting into the nucleus, and I said we're focusing on important, so there are other proteins that help get into the nucleus, and then we talked about exportant, and there are other proteins, and the book has a little table of them, because there would be these other examples. There are a variety of examples of the things that are needing to get in and out. And sometimes it's not just like an individual protein that needs to get in, it's like a assembled thing. And so that's what's leaving. So the ribosomal RNA doesn't get to go out unless it's part of a finished subunit. And yep.
Are there any importance and exportance from the nucleolus, specifically?
No, again, the book artist depicts these factories and nucleoli as having a boundary. They don't. There's no membrane, there's no gate physically around them. It's just a place where it's more concentrated, like that, And, you can think about a normal analogy, right? Where, oh, wow, look at that flock of crows. They're all together there, but there's no boundary on those crows. They're all there because there's food there. It's like that. No physical boundary to get out of the nucleolus, because it's just a place where a lot of stuff is happening, like that. Yeah.
So Professor, you say the ribosome RNA, it's not leaving, it's staying in the nucleus?
Yeah, I'm pretty sure that's true.
Okay, what is leaving? The ribosome protein?
Yeah, so let's just talk about that again, because what's happening is the ribosomes are being assembled in the nucleolus, and the ribosomes are made up of RNA, ribosomal RNA, and ribosomal proteins. So where are those made? Where are those coming from, right? Like that, right? So for the ribosomal RNA, that genes are in the nucleus. And it is, and I'll whisper this, a transcription factory, right? Because that's happening there. They're making the ribosomal RNA there, and the ribosomal RNA is getting processed there, getting ready. It has to get cut up a little bit and modified a little bit. Now, what about those ribosomal proteins? And that's, I mentioned that, but it's good to talk through that again. Because those are proteins that are made by the genes, and those genes are not in the nucleus. The genes for the ribosomal protein are scattered around inside the nucleus, like all the other genes we talked about in our course. And they'll be in a ribosome factory, I'll say that aloud, because they are somewhere else in one of these ribosome factories. And they're getting expressed just like the genes that we've studied, right? They gotta have something bind to their enhancer and activate transcription, and we get the primary transcript, and then it's gonna get the RNA processing, and described in that handout. And they're just like any other protein. Then the mature mRNA leaves the nucleus, and it binds to an existing ribosome in the cytoplasm, and it makes that one ribosomal protein, of which there are 7 different ones. Then that one has to have a nuclear localization signal, because it has to get back in, because it's got to find its way to the nucleus. And so that's what's happening there, is that again, we're assembling the ribosomes, and the ribosomal RNA are there already. The ribosomal proteins need to come there, like that. They have their genes scattered elsewhere in the nucleus, and those have to get transcribed in a transcription factory and processed and out into the cytoplasm, translated into a ribosomal protein, ready to go, and they have to come back in and come to the nucleus for assembly there. Another way to say that is when we are making more ribosomal proteins, because we need more ribosomes, those proteins don't just join existing ribosomes in the cytoplasm. They need to go back into the nucleus and get assembled into ribosomes. You might say, why don't they just join a ribosome? Let's go. They get made by a ribosome, they could just be making, like when they do it, they don't. It's gotta get assembled in the nucleus. So that's that path of the story, yeah. Yep, other questions people have?
Yeah, I got a question
Yeah.
About 1433. So you said you can bind a protein and then cover that protein so that that protein can get the nuclear localization signal.
Yeah.
And then out to the cytoplasm. So
so the 1433 would be binding it in the cytoplasm and uncovering a nuclear localization signal when that protein's in the cytoplasm, and that's allowing it to go in through the nuclear pore to get into the nucleus.
So 1433 is going to do it for all the proteins, or just a specific protein that 1433 binds to?
Just specific proteins, yeah. And there are many proteins in that 1433 interactome. And almost all of them are like FOXO, where 1433 binds to the phosphorylated form only, and it covers the nuclear localization signal, and it sequesters, it keeps the target in the cytoplasm.
So we can have another protein that does the same job than 1433.
Oh yeah, I mean, I don't know of them, but I'm sure there are other proteins that play that role. Yep. Yep.
What releases the binding of 1433? Like, is it a dephosphorylation?
Yeah.
Or-
Yeah, I don't know. That's somebody would say, their kind of educated guess was, what I kind of talked about is you gotta remove that phosphate. But there might be other mechanisms. I don't know. Yep.
And then, when transcription factors get phosphorylated, some get turned on or off, does the binding of 1433, like, when one of them is turned on, like FOXO is turned off when it's phosphorylated, but like
So that's, yeah, so we've seen lots and lots of proteins get phosphorylated. Almost none of them bind to 1433. So even though 1433 does a lot of different things, there are way more proteins that get phosphorylated than are in that 1433 interactome. And I don't know for sure, but like almost none of the phosphorylated proteins that we talked about in our class are regulated by 1433. Some are, you know, but you would say, nope, they're just phosphorylated. And, you know, in the last minute, we have 4 minutes left, but, you know, last 2 minutes. So it's a really interesting thing. So let's just talk about this too, because it goes back to that information story and the idea of ATP. So what's going on is that, so let's pick a different protein that I'm pretty sure is not regulated by 1433. Let's say elk. And so here's elk, and you know, elk would have some sort of shape like that. And it turns out that elk, as it's just hanging out, can kind of flip between two shapes, like that. Just its structure is built so it can flip between two shapes, kind of like a teeter-totter, going between two shapes, like that. And that could just happen randomly because it has a kind of flexibility in the protein. And so it's got these two conformations that could be in. And this is inactive, and this is active. And one of those shapes is more common by far. So it could be in both shapes, but one is more common by far. And for elk, this shape is going to be more common. It's just sitting there like that. So it's like a teeter-totter with a heavy kid on one end. It's just sitting there. It's not going to go back that way. It could. It's not going to. Like that. Okay? And so then we have ERK. And ERK gets phosphorylated, gets activated. Here comes ERK. And ERK's job is to add a phosphate to elk. So ERK is a kinase. And the kinase has that substrate, right? It's going to add a phosphate. Where these kinases all get the phosphate is from ATP. So the substrate for ERK is elk plus ATP, and the products are phosphorylated elk and ADP. So it's burning ATP, it's using energy. And what it's doing is, it turns out that the protein elk is built so that when it's phosphorylated, that's the common form. So it's literally like that thing where you have the heavy kid on one end of the teeter-totter, and you go and stand. ATP uses energy to stand and flip it over. So it's much more common. And that different shape is going to be like, that's maybe whatever, something, the DNA binding domain or something like that. You know, it's somehow changed its shape, so now it does its job. And you're using energy just like the freaking light switch, you know, where the light switch is off. And the light switch is not floppy, it's like really kind of stuck off, but it could get on if you use a little energy to kick it over. And then you kicked it over, and now that's the real stable one, you know, like that. So the deal is, we've seen, right, that sometimes the phosphorylated form is active, sometimes it's the unphosphorylated form. It's just whichever form is more stable, you know, like that. That's the switch, it's always that. You know, it's always that one form is more stable, and you phosphorylate it to shift it over to the other form. And it is just, you need energy to click it over, push it over, because it could go there, it's not going, because it's just not, you know, as stable unless you phosphorylate it. Then it kicks over. And that's why, you know, there are 600 different kinases in humans like that. You know, it's just such a good way to do post-translational regulation by post-translational modification. You make the covalent change, and it's just on, I'll be on, I'll be on, I'll be on, I'll be on all day. I'm not turning off till you remove that phosphate, and then I just fall back over to the on-state of the inactive form. Yeah, just like, wow, man, super, you know, evolution should take advantage of that, and then it did. Yep, we have to stop. But yeah, it's amazing, right? We should be going getting tattoos right now.